2 hour Saturday live study. I like to say live lecture. Uh, By a show of hands, who likes that term lecture? No, Jonathan's like, come on, man, make it to yes, lecture. Me personally, I just kind of like it. But who does not like that term? Raise your hand if you don't like it. Rasheen, what's up? Destiny, what you guys do? Is it too academic-y? Is that the problem? Yeah, okay.
When I hear the word lecture, it's like somebody's about to give me a rundown of a whole bunch of information that I might remember like 10% of, and it just sounds overwhelming.
That's what Corey said to me. He said, I don't know, man. It's just when you use the word lecture in there, and I'm like, no, lecture's cool because I love going to lectures. But there it is. I'm an academic, so I like that. But if you don't like it, I'm going to take it off of the inside the portal. I have live lecture study, so I just want to make sure that everybody's good with it. So, but yeah, welcome, guys. Remember, these do get recorded. They go into the Facebook. Well, they go into Facebook after I upload them into YouTube, just so you guys know. So if you ever want to go back and look at them, you're more than welcome to. So before I open up the workbook, does anybody have any questions either from last week, what we went over? Remember, we talked about muscle spindle fibers, Golgi tendon organs, things like that. Remember, does anybody have any like burning question from that from last week? Yay, going once, going twice. Okay. Well, there you go. Guys, let's go ahead and get started. We are going to jump right back into it. I'm going to go ahead and open this up for you. Give me a thumbs up if you can see the screen. Yay. Yes. Okay. So if you remember, we were in module 1, lesson 1. We ended talking about the muscle spindle fibers. Remember, if you have any questions as we are going through this, please either raise your hand or throw a message or a question in the chat. Please stop me at some point. The goal of doing this was to allow for questions sort of on the fly as we're going through the materials. Does that make sense? My recommendation is have a pen, pencil, whatever, taking notes either on a sheet of paper or in your workbook itself. Does that make sense? Okay. Get Miss Queen in here. You know, we might actually, by the end of this, have enough people to get a 4x4 picture. So I might do that. Okay. So this is where we left off. We now move into another really important area. And remember, folks, for the most part, the materials that we provide you in the BDU modules is, I put it in air quotes, real world material. In other words, you're not going to talk to clients about you're not going to use certain terminology. You do have to know certain terminology to make it through the academic part of what we do. Does that make sense? So you got to know but once we get into an area like muscle fiber typing, we're talking about real world application. This is literally what we use. When I used to do athletic training and worked with athletes, this was the main way we determined not only how to make somebody faster, but how to do the training. This is the real world when we get to it. You all with me on this? So when we talk about muscle fiber typing, there are 3 types of muscle fibers. Does anybody know what they are, by the way? Does anybody? Just go ahead and yell it out if you want. Unmute yourself and yell it out. What are the 3 types of muscle fiber in skeletal muscle tissue? Come on. Questions get a lot harder from here.
Type 1, type 2A, and 2X.
What, Francette, was that you?
Yeah.
Give her a hand. Yes. Yes. Come on. Great job. Yes. By the way, there's actually, I think at last count, there's actually 6. But now we're digging so deep below the surface. We have 3 main functional type of muscle fibers: type 1, type 2A, and type 2X. We generally start off with the designation type 1, type 2, right? Slow twitch, fast twitch. You guys have heard these terms probably somewhere along the line. What we do is we then subdivide the type 2 fibers into what Francette just explained, type 2A, and then type 2X. So we have type 1, which are clearly designated all on their own because of their basic metabolic attributes. And then the type 2 fibers, which are what we call fast twitch. And those are actually, there's actually multiple types of fast twitch, but the two main ones that we normally deal with because of the overall total percentage, they make up the largest percentage of fibers in the average human, is going to be the type 2A and the type 2X. These are going to tell you a little bit more from the metabolic attributes component. Okay? Is everybody with me so far? 3 types of muscle fibers.
Yeah.
Type 1, type 2A, type 2X. Okay. The first one is the type 1 muscle fiber. Now, remember, when we designate fiber typing, we're looking at a number of different components or variables that tell us what these fibers are able to do or what they do inside an actual muscle belly. The type 1 muscle fibers are what are known as, and there's a bunch of different designations. Try to do your best to memorize this, okay? They are what we call slow twitch or slow twitch oxidative. Now, one of the things that can help when it comes to memorizing these different muscle fiber types is remember that slow twitch muscle fibers are going to have a very high endurance capability. If you look at the word oxidative, what's the first word that comes into your brain when you think oxidative? What's the word? Oxygen. Oxygen, right? Maybe. Well, if you didn't think about that, that's what I want you to think about, oxygen, right? So slow twitch oxidative muscle fibers have a number of different attributes associated with them. Okay. They are red. Now, and by the way, when I say they're red, remember what we talked about? Muscle cells, muscle fibers are microscopic. So you got to look at these things under a microscope. And what researchers do when they're looking and studying muscle fibers is they'll take a plug generally, or if they're using cadavers or something like that, and they'll look at the fibers under a microscope and they will stain them with a phenylalanine-based type of stain. And certain organelles inside the cells will do what we call take the stain. Any of you that have how many of you have been in like a biology class where you added the phenylalanine right in the it's kind of purple, right? Okay, so you put this stain on there, and it just so happens that mitochondria and certain organelles will take that stain, and they will appear red, purplish red, under a microscope. You guys got that? That's why we call them red, because they contain organelles or components in the cell itself that take a stain, and then it appears red. Got it? So now watch what I do from a studying perspective. Slow equals, I like to use equal signs, just so you all know. You can use whatever you want, but I always say equals, meaning this is the definition. You can use an arrow. I use an equal sign. So slow equals oxidative, in parentheses, oxygen, / red. Does that make sense? And then I start listing the other attributes. The next attribute that you see here is low power output, meaning that these muscle fibers do not have the high level of power generation that the other muscle fibers do. Does everybody remember what a muscle fiber is made up of? Does everybody remember? Muscle fibers are made up of myofibrils.
Myofibrils.
I'm going next into that. What are myofibrils made up of? Sarcomeres. What are sarcomeres made up of? Now you can say it. Actin and myosin filaments. You get that?
Correct.
So if you have a muscle cell that for whatever reason does not have as many myofibrils in it, it's simply not going to be able to generate high levels of power output. You got it? What generates power or tension in muscle fibers? It is the, guys, stop me if I'm going too deep on this. It is the cross bridging that occurs between the myosin and actin filaments. So when myosin heads attach to the actin filaments, the proteins inside the sarcomere, they attach and essentially hold on to it. That creates tension. So now the sarcomere can create tension. Got it? If I have enough cross bridges within enough of the sarcomeres, within enough of the myofibrils, the entire muscle cell can actually create tension and shorten. That is a single cell I'm talking, single muscle fiber that I'm talking about. The only reason I'm saying this is because I want you to understand why type 1 muscle fibers have these attributes that I'm going over. Does this make sense to everybody? I just have to, sometimes I go pretty quick on this because if you study it for 50 years, it just makes sense. If it's not, then please stop me. Okay?
It's clear
So-
as mud.
Say again, clear as mud. Right, exactly. So low power output. So I put slow twitch equals oxidative oxygen, red, low. I don't write power output, by the way. I just write the word low. I'm trying to study this and memorize it. So I'm not going to write every single word out. What's another attribute? They are small, relatively small in diameter. Now, if a muscle cell is small in diameter, it goes back to what I just previously said. It probably doesn't have as many myofibril components to it as a larger muscle cell. Is everybody with me? Yes? So type 1 slow twitch muscle fibers tend to be, folks, I said tend to be smaller in diameter than their cousins, the type 2A and 2X fibers. Now, they are also, because they are low in power, there's a reason they're low in power, and the reason is because of the myofibrillar content, reduced number of potential cross bridges, but guess what? Type 1 muscle fibers are very high in their endurance. They are aerobic. Aerobic means with oxygen. They have high endurance capabilities. Can anybody tell me why they have high? And by the way, when I say they have high endurance, that means that the muscle cell is able to shorten, lengthen, shorten, lengthen, shorten. It's like the Energizer Bunny. Boop, 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 boop. And it can do that for an extended period of time. Does anybody know why type 1 muscle fibers have such high levels of endurance? Does anybody know?
Is it because of the mitochondria?
Who's just said that? Mimi.
Mitochondria, the powerhouse of the cell, right?
Give Mimi a hand. Exactly. Yes.
Thanks.
Mimi, that's exactly right. The mitochondria, as you learned in ninth grade, well, those of you that actually went to class that day, ninth grade biology, Helen, right? Helen's like, yeah, I didn't make it to biology that often. If you were there and you were awake, you learned when your biology teacher drew this circle. And by the way, I don't know any cell in the human body that's actually a circle, but they drew a circle, and then what else did they draw? They drew a circle inside a circle, and that was the nucleus, right? Am I bringing back bad memories? Should I stop? Right, from high school? And your teacher probably drew all these little, what are known as organelles. The human cell, every cell in nature is an incredibly complex factory. Okay. And these organelles all have different things that they do. One of the organelles is what provides long-term energy. See the word long-term energy for the cell. And it's known as a mitochondria. So yes, Mimi used the term, the quote, powerhouse of the cell. However, are certain cells in your body capable of getting energy and producing energy and doing all the things they need to do without mitochondria? Yes or no? Let me give you a hint. Yes. They are called type 2X muscle fibers. Mitochondria are incredibly valuable because they provide the vast majority of ATP production or energy production within the vast majority of cells in the human body. When we look at a type 1 muscle fiber, they are by design, loaded with mitochondria. I don't want to get too deep into it. We'll talk about it at a later time. But mitochondria basically create what's known as, or the electron transport chain is what we find within the mitochondria of the cell. We'll talk about that at I think lesson, maybe the next lesson. But the point is that type 1 muscle fibers have a pretty good amount of mitochondria in them. Therefore, they can generate ATP continuously. In a sense, as long as there's enough substrate, i.e. glucose, free fatty acids, even amino acids, certain amino acids, as long as there's enough substrate in the cell, type 1 muscle fibers can essentially contract forever. In a sense, they could. Okay, as long as there's enough substrate and enough of the material, enough of the waste materials, carbon dioxide is being eliminated from the cell, type 1 muscle fibers can keep going, like the Energizer Bunny. Is everybody with me on this? That's why they have almost unlimited endurance. Got it? Everybody is sitting down right now, yes? Now, your muscles, the muscles that are keeping you erect sitting in your chair, what muscles are responsible for that are keeping you erect? They're located in the lower part of your lumbar spine, and they're called the what? Erector spinae muscles. Okay? Those muscles right now, every one of you right now, those muscles are contracted, aren't they? They have to be. They're keeping you upright. But those muscles, the muscle fibers inside those particular muscles, they have to keep you like this for how long? Minutes? In fact, hours in some cases. You can stand up for a long time, can't you? So your body needs, or your body has to have muscle fibers that are able to contract continuously. Very low grade contractions, right? To keep you in, posturally sound positions. Is everybody with me on that? Got it? So your body has these muscle fibers that are designed to just stay contracted, or moderate levels of contraction, more than tonus, by the way, more than tonus, but to keep the body in positions of posture generally. That comes from the type 1 muscle fibers. Is everybody got this? And what I should have told you beforehand is that every muscle, every skeletal muscle in your body has all 3 types of those muscle fibers, just so you know. So your erector spinae do have type 2A and type 2X fibers in them. But right now, the vast majority of muscular activity that is occurring in the erector spinae muscles of your bodies right now is predominantly type 1 muscle fiber usage. In fact, I would venture to guess that the type 2 muscle fibers, both 2A and for sure the type 2X muscle fibers, they're on vacation. They're just relaxed. They're not even working, more than likely. Does that make sense? So when you go out to take a walk, guys, when you go out and you just walk at a very comfortable pace, What is the predominant type of muscle fibers that are being used in the muscles of propulsion?
Slow twitch.
Generally type 1 muscle fibers. There might be some type 2A in there. But the point is that these muscle fibers are designed for endurance. And how long can you walk for? 10 minutes? An hour? Some of you 2 hours? You get what I'm saying? So keep that in mind. Type 1 muscle fibers are responsible for postural control because they're needed all the time. They cannot tire out. Got it? Any questions on that? Speak now or forever hold your peace. So type 1, characterized by relatively low force. You guys can read through this. Mitochondrial density, very high, responsible for the general muscle tonus and lower level contractions, found in higher percentages in the postural muscles. Okay, now you have your fast twitch muscle fibers. There are generally 2 types, what we call type 2A and type 2X. The type 2A are what are known as oxidative, very similar to type 1, right? Oxidative / glycolytic. What does glycolytic mean? Anybody? What's glycolytic mean?
Has to do with glucose, right?
Keep going, Hannah.
The use of glucose. The glycogen stores uses glucose for the power.
You're right there. That's pretty good. So guess what? Give her a hand. Give her a hand. Yes. When we say glycolytic, we are talking about anaerobic glycolysis. And yes, anaerobic glycolysis begins the ATP production process using glucose. Got it? So the point here, folks, is that there's a lot of terms that I'm throwing out at you. Remember, you are studying in conjunction with what we do here on Saturdays. I want to make sure you write some of these words down. Do you know what the difference between aerobic and anaerobic is? I see people get these questions wrong on the final exam all the time, right? Aerobic with oxygen, anaerobic without. So when we say oxidative, we are talking about aerobic processes for the production of ATP. And ATP we'll get into within another couple of lessons, okay? Anaerobic means without oxygen. So therefore, all right, who's the smarty here? If we are producing, in this case ATP, without oxygen, what organelle do you not find in those type of muscle fibers, type 2X muscle fibers? What type of organelles do we not find because they're not endurance fibers? Mimi?
Mitochondria.
Mitochondria. Does everybody get it? Mitochondria is what allows the stain to look purplish red under a microscope. So the more mitochondria you have, the redder. I use air quotes a lot. The red is redder a word, by the way? Does anybody know? If it's not, it is now.
The gnome color.
It's what? It's redder. Okay? So the deeper the red or the deeper the stain, probably the greater the level of mitochondrial density inside that type of muscle fiber. So guess what we have? We have what are known as type 2A, which we call pink fibers. Everybody see that? Why are they pink? Because they're not as red as type 1 fibers, which means they don't have as much or as many mitochondria. So therefore, if I take type 2A muscle fibers and I stain them under a microscope, they don't have the same mitochondrial density as type 1s. So therefore, they don't take as much stain. They look less red, hence the term. You'll see this in some books, textbooks, pink. You get it? So we can look at these muscle fibers and say certain muscle fibers under a microscope that, and they've been stained, some of them look red, some of them look not as red. They kind of look pinkish. And then others look white. They don't take the stain at all. And those are your type 2X fibers. Is everybody with me? So type 2 fibers, both 2A and 2X fibers, are what we call fast twitch. Now, not today, but next week when we do this, I'll explain the neurological components, why we call them both fast and slow twitch. But for now, just for today, I want you to get these concepts of the metabolic attributes, right? So they're pink in color, moderate in power, which means that they have a higher level of force output than the type 1 fibers, but not the same level of force production as the type 2X. So on a continuum, type 1, type 2A, type 2X, we have low power, moderate, high power output. Does that make sense? Now, the type 2A fibers have some endurance components to them because they do contain mitochondria, so they can actually contract, shorten, and lengthen for extended periods of time, way longer than just the type 2X fibers, okay? So type 2A, oxidative glycolytic, just showing that it's kind of halfway in between, right? Because type 2A fibers are not purely oxidative, they are also glycolytic, which means that they actually have anaerobic glycolysis as a method of producing ATP inside the cell. Got it? Type 1 fibers, for the most part, they don't have any they don't need any glycolytic component to the ATP production process. Type 2A has both. It's kind of like a what are those cars that use gasoline and electric? What do they call them? Hybrids, right? So type 2A is kind of like a hybrid. And then type 2X are essentially purely, there are some genetic modifications, differences from one person to the next. But type 2X muscle fibers are purely glycolytic. They have little to no mitochondria. Therefore, even if there is a very small amount of oxidative phosphorylation occurring inside the mitochondria, it is so minor, it's just not worth talking about. So we say that type 2X fibers are purely glycolytic, and they function, ATP production function through the glycolytic or anaerobic glycolysis. Does that get that? Now, again, it's the terminology that you do need to get. Who's got questions on this? Rasheen, did you just yawn while I was talking? I'm just kidding. Who's got questions on this? By the way, if I know the deal. If you're confused, I wouldn't be surprised. I'm hoping that when we do the live lecture, that it makes a little bit more sense. But remember, even if you do have questions after this, what can you do? Yeah, you can ask me questions, right? Yeah? So if you do have questions, even from what we just went over, shoot me messages in the comment box. Okay, let me see. No, I don't. I needed 3 more people, so I can take a picture. Folks, is this helpful today? Yes? Please make sure, because this is going to it's recorded and it's going to get posted into our YouTube, and then it's going to get put into Facebook as well. So if you do want to see a recording, the copy of it, you're more than welcome to do so. Any other questions before I let you all go?
Yeah.
Terry.
I wanted to know, I went to schedule for the 23rd. Are we not doing it on the 23rd?
Is it not open?
It shows the 16th and then the 30th.
I will go in there and see what's up. But yes, it should be. Just check in the next day or two.
Okay.
Okay, I'll take care of that. Okay, guys. Have a great weekend. It was a pleasure. Y'all take care, and we'll see you next week. See you.
Thank you.
Thank you.
Thank you.
Bye.